Hocam isteği kabul eder misiniz? Mr. Khawar, can you accept our Mahmood request?
Yapabildim mi?
Tamam hocam.
Tamam. Şimdi ben başlıyorum.
Buyurun hocam, başlayın.
Distinguished ladies and gentlemen, the topic of our discussion is post-germination seedling growth performance of two safflower cultivars, Dinçer and Remzibey, against drought and salt stress tolerance. The presentation is jointly prepared by Fatma Kayaçetin and Khalıd Mahmood Khawar, my name. It is well known that safflower is counted among the oldest known crop plant species. It is adapted to hot and dry environments. Safflower is self-pollinated crop and diploid and has deep-rooted system. It is branched and an annual plant. Turkey has produced about 21,883 tons of seed safflower on about 15,816 hectares during 2019. Safflower is more resistant to high temperatures compared to other oilseed crops. It is one of the promising and outstanding oilseed crops since its seed oil is about 32 to 40% that contain about 90% unsaturated fatty acids. These fatty acids are mainly composed of oleic and linoleic acids. Safflower stigmas are commonly used for dyeing, flavoring, and various medicinal purposes. These stigmas are commonly called pseudo-saffron in many East European countries and Turkey. Drought and salinity are two major environmental and stress parameters that limit plant development in all semi-arid regions everywhere in the world. Seed germination percentage and developing during early stages of growth are highly affected by salt and drought stress. The objective of the study was to investigate these two parameters using PEG and sodium chloride on cultivar Dinçer, that is spineless variety, and cultivar Remzibey, that is spiny variety. The results of the study would be exploited in future safflower breeding programs of Agricultural Research Institute, Yeni Mahalle, Ankara. The study was carried out at the same institute as I mentioned earlier, Central Research Institute for Field Crops and Ankara University. The major factors, the two experiments were conducted. One experiment had main varieties, Dinçer and Remzibey as main factor, and sub-factor was different doses of PEG, as shown in this slide. The second experiment also had Khalıd Dinçer and Remzibey as main factors and different levels of sodium chloride as sub-factor. Water was used as control treatment in both experiments. 3 replications of 20 seeds were used in each petri dish and were germinated in 2 layers of Wattman qualitative filter paper grade 1. The filter papers were replaced every 2 days in new petri dishes to prevent accumulation of respective PEG and sodium chloride solutions, and also to stop development of fungus. Germination percentage was recorded on daily basis till 10th day of the experiments in accordance with ISTA rules 1996. The seeds under both stress conditions in both experiments were considered germinated when approximately 2 millimeters of radicle emerged from the seeds. Thereafter 10th day, the final germination percentage, root length, shoot length, seedling fresh and dry weights were measured. The dry weights of the sampled seedlings were measured by taking the fresh weights of the seedlings to oven at 70 degrees centigrade for 48 hours. Analysis of variance for all investigated parameters was performed using MStatC. The variations among the parameters were carefully compared by LSD, or least significant difference test, at significance level of 0.05. The effect of sodium chloride and PEG on final germination percentage and other parameters taken in the study are described in the next slides. First, I will describe the experiment about PEG, or the experiment about drought stress. The experimental results showed that different levels of PEG concentrations induce significantly different effects on final germination percentage and other parameters taken into consideration in this study. The results showed that each increase in the concentration of PEG induced increased osmotic stress with related decrease in germination, seedling growth, and related parameters such that minimum stress was noted on the minimum stress level of, and the maximum stress was noted at the maximum dose employed during germination treatments. I will show these in figure in the next slides. The results showed that maximum germination percentage of 91.11% was noted using 0.2 megapascal PEG 6000 concentration. The maximum root length of 4.3 centimeters was observed for cultivar Dinçer using control. The minimum root length was detected for cultivar Dinçer using -1 megapixel PEG strength. These results are better explained in this figure, where a maximum seedling length is shown under controlled conditions when the seeds were germinated using water. At -0.2 megapascal PEG strength, the seedlings induce inhibition in both Remzibey and Dinçer cultivars. This inhibition was more pronounced in cultivar Dinçer compared to cultivar Remzibey. This inhibition was more pronounced and more depressing at concentrations of -0.6 to -1.4 megapascal. These inhibitory effects and reduction in germination are highly pronounced at -1.4 megapascals as shown in this slide. This showed that reduction of water potential using PEG induced a significant decrease in fresh and dry weights of seedlings. Statistically significant differences were found for germination percentage and all related seedling growth parameters using sodium chloride stress, different levels of sodium chloride. Increased levels of sodium chloride stress showed increased stress on seedlings. However, seedling stress levels showed that the performance was better compared to drought stress. In both experiments are independent experiments. No experiments use both stresses together. We see minimum stress at 5 decisiemens per meter sodium chloride stress when we use this level of sodium stress. And this stress, the seedlings that develop at this stress are almost of same growth and same stature that we grow using water. This means that the safflower seedlings can can bear this stress. However, thereafter, we can see a minute depression in growth of a cultivar Remzibey compared to cultivar Dinçer. This stress was pronounced at 20 decisiemens per meter in both cultivars. After 20 decisiemens, the seedlings are showing complete or contrasting contrasting depression in seedling growth. Both seedlings, both types of cultivars show complete stress or stoppage in growth at 35 decisiemens sodium chloride stress. These I will like to describe shortly these in terms of various growth parameters in next slides. We had 93.3% are close by seed germination on 5 decisiemens here. Minimum germination was noted here at 30 decisiemens in cultivar Dinçer. The maximum seedling fresh weight of 2.99 in cultivar Remzibey was noted at 0.2 MPa megapascal and cultivar Dinçer. The minimum fresh seedling weight and seedling dry weight were observed for Dinçer. Here. The seed germination percentage of both cultivars showed a complete inhibition at 1.5 megapascals or minus 1.4 decisiemens. In conclusion, we can say that present study compared different doses of drought stress using PEG and sodium chloride salts using sodium chloride stress, and these showed adverse effects on seedling germination during early stages of growth. These stress levels can be recommended and can be used for breeding purposes. This can be recommended to the farmers that they can grow their crops on these levels of stress if these levels of stress are there on their lands. Otherwise, there will be significant drop in their crops. The best, if we compare the results, the best tolerant genotypes, we find that Remzibey was comparatively more resistant compared to cultivar Dinçer for drought stress. And cultivar Dinçer was highly and more resistance compared to cultivar Dinçer, cultivar Remzibey for salt stress. I'm highly thankful to you, my distinguished friends and colleagues. My presentation, and this is the end of my presentation. Thank you.